Thank you very much, and thank you for the opportunity to talk to you here today. So I set myself a really modest goal in this talk, which is basically to ask the question of how do genes influence phenotypic variation? And I probably should have given myself a little bit of a reality check when I realized I was going to be talking to a room full of population and quantitative geneticists, but nonetheless, here we are. So we'll see how it goes. So how do genes influence phenotypic variation? Well, the kinds of answers that you work on here are answers of a certain kind. They're statistical answers. So genetics provides a statistical relationship between phenotypic variation on the one hand and genetic variation on the other. So for highly polygenic traits, as many of you are probably encountering, these kinds of answers can be dissatisfying. They can be dissatisfying because they essentially consist of very, very long lists of genes that may be somewhat problematic in terms of their association with each other. So this is the frustration of modern day genome-wide association studies that I think we're all familiar with. Developmental biology provides a different kind of answer to this question. So developmental biology is in fact not really concerned with explaining variation. It's concerned with explaining mechanisms, understanding pathways, how genes act together to do things in development. And so variation is something that is an afterthought or sometimes not even within the frame for developmental biology. In fact, the mechanisms that underlie phenotypic variation are often lost in translation between genetics on the one hand and developmental biology on the other. And when you think about it in those terms, you realize that neither field tends to ask this question of how variation acts on mechanisms in development to produce differences among individuals. What are the mechanistic bases for phenotypic variation? Is a question that often falls in between the cracks between those people who study variation explicitly, geneticists study variation explicitly, but they do so from a statistical point of view, and developmental biologists that study organismal structure or the genesis of organismal structure from a mechanistic pathway developmental mechanisms point of view. So one problem with this question is that the ways in which genes influence phenotypic variation is really complicated. So genes act through multiple levels of things in organismal development to produce their phenotypic effects. So if you think, for example, of a gene that might influence the shape of the skull, this is something that we study in the lab, you might imagine that there'd be some sequence variation in a gene or regulatory site, like in FGF8, for example, that then produces variation in either expression of that gene or in the function of that gene, that in turn then produces variation in some cellular process, some aspect of cellular dynamics, proliferation, polarity, interaction with extracellular matrix, all kinds of things can be influenced by this genetic variation. That in turn produces some variation in morphogenesis, which in turn produces variation in morphology, in the sense that the tissues that are affected in morphogenesis here ultimately become something that resembles an adult organism. And so there are multiple levels of process through which genes act in order to produce their phenotypic effects. It makes it complicated. Genetic disease, in some ways, is just genetic variation that is deleterious. And it's deleterious in the context in humans that results in the label of a genetic disease. So these are heritable diseases associated with genetic variation and associated with deleterious effects in humans. We can think of genetic disease as forming a continuum. You know, the standard account is that we divide genetic disease into Mendelian disorders on the one hand and complex disorders on the other. But in reality, it's much more complicated than that. In fact, there's a continuum from diseases that tend to be highly penetrant and are associated with one or a few mutations, and diseases that are highly complex, like diabetes or obesity or hypertension. And this is complicated on a number of different levels. Even for diseases that are monogenic, penetrance is often highly variable. So this is frequent, in fact, probably the norm rather than the exception, that genetic diseases in humans that are within this category of Mendelian disorders have this feature of varying significantly in both expressivity and penetrance. This, for example, is a panel of images from Max Munke at NIH showing individuals who have the same mutation in sonic hedgehog. And they range in phenotypic expression from embryonic lethal forms, of cyclopia here, to a phenotypically normal person down here. All of these people have the same mutation. It's a supposedly highly penetrant Mendelian disorder. The other thing is that as we're starting to sequence more and more individuals in the background population, this is coming out of things like the 10,000 Genomes Project, we're finding more and more individuals that have these supposedly highly penetrant mutations, but don't have any visible disease. So they were enrolled in these studies because they thought they were phenotypically normal, but in fact they have some highly penetrant disorder. And which I think highlights the ascertainment bias that we often have in the detection of these diseases. People normally get genotyped for a genetic disease because they have the phenotype. And so that misses all the people who might be out there in the population who also have the same mutation, but don't have the phenotype. It's also complicated because of interaction effects. Context matters. Epistasis is very common. Here's an example of work that we did a few years ago. We wanted to see whether different sprouty mutants would tend to have the same effect when we put them on different backgrounds of mice. Turns out not only were the effects of these 3 sprouty mutations different, they sometimes were opposite in direction. So these are mutations of large effect. And sometimes, in the case here of a sprouty 2 on a C57 background and a sprouty 2 on a 129 background, the craniofacial shape effect, exactly the opposite. So major mutation having different direction of effect depending on genetic background. Epistatic interaction is very common. Context matters in genetics. Now, within evolutionary biology and evolutionary developmental biology, people spend a lot of time thinking about how to make sense of these kinds of complexities. And so there's a variety of heuristics that I would group under the general heading of genotype-phenotype maps that have come up in order for people to make sense of this stuff. Ways of incorporating development into our thinking about how genes might influence phenotypic variation. Here are some examples of these genotype-phenotype maps. I mean, the first one I show here actually it doesn't involve development at all. This is Lewinton. It should be familiar to most of you. This is actually one of the earliest genotype-phenotype maps, although one could argue that Wright's 1934 gene expression or gene function to phenotype curve is also a genotype-phenotype map of a certain kind. It differs from Lewinton's map in that Wright actually did incorporate development into his thinking, if you read the associated text. And here are other representations. This is Günther Wagner and Altenberg showing epistasis and pleiotropy forming modular patterns of organization and gene effects on phenotypes. Here's one from me showing how one might think about developmental processes, funneling genetic variation into sets of correlated traits. There are multiple ways of doing this. Here, of course, Waddington's epigenetic landscape is a form of a genotype-phenotype map. These are not real things. They're heuristics, ways of trying to make sense of this underlying complexity of how genes act through developmental processes to produce phenotypic variation. Okay. So let's take as an example variation in the face. So we're going to talk about the genetics of the human face, but I'm also going to talk about the genetics of craniofacial variation in mice and try and relate them together. So facial shape is highly heritable. You've all seen this within your families, right? Here are some examples of famous people because we can use their images for free, showing the resemblance between fathers and sons in this case. These are remarkable patterns of resemblance between relatives, particularly when you take into account how extraordinarily polygenic the face is and how extraordinarily small the individual gene effects that have been associated with facial shape are. The particular patterns of resemblance are remarkable in light of that fact. So facial shape is highly heritable. There's a heritability of around 0.7, depending on the population. But it's determined by a very large number of genes, extraordinarily polygenic, perhaps thousands of genes. It may be like stature, a trait that sort of borders on the omnigene model. And I say that because facial shape is at least as complicated as stature, because it turns out the stature is correlated with the shape of the face. So anything that affects stature is going to have some small effect also on the shape of the face. These are genes that have been associated with facial shape. Together, they explain 13.7% of the total variance, total heritable variance in facial shape as of last year. And I hate to think of the amount of money that's gone into the genome-wide association studies that have produced these genotype-phenotype associations, but it begs the question of whether continuing in that project is worthwhile, because ultimately we're just going to end up with a longer and longer list of genes. I'm not sure that that list will make us understand the genetic basis or the heritable or the mechanistic basis for the heritable variation in facial shape any better. Okay. So one thing that we've studied in my group over the last few years is the relationship between facial shape and genetic disease. And this may seem like kind of an odd thing to do, but if you ask a medical geneticist, it seems like a really obvious thing to do. Because in fact, medical geneticists have been using facial features to diagnose genetic disease for a very long time. In some ways, it's a human interaction type of thing. A patient walks into a clinic, and as you know, we're humans. The first thing we notice about each other is our faces. And so it's normal that facial features that are associated with genetic disease might become diagnostic criteria for genetic disease, because medical geneticists have been doing this for a very long time. But why does this work? Why on earth would it be the case that thousands of different genetic diseases might map in some coherent way onto variation in the shape of the face? And I think that's the remarkable thing. I don't know the full answer to that question, or the answer to the question of whether facial shape is unique in this regard. If we all had X-ray vision and we could look at the three-dimensional structure of our kidneys, would we be able to diagnose genetic disease from each other's kidneys as well? Possibly. I don't know the answer to that question. But I think that there are three contributing factors for why facial shape variation works as a diagnostic tool for genetic disease. And one of them is, as I was saying earlier, facial shape is heritable and extraordinarily polygenic. The fact that it's so polygenic creates a lot of potential for genes to have an effect on the face, right? There are thousands of genes involved in the face. Presumably, a disease involving any one of those genes could have some effect on the shape of the face. The second answer is pleiotropy, because most of these diseases are not diseases of the face or even diseases of craniofacial variation. They're not craniofacial malformations. They're often metabolic diseases or connective tissue disorders. And the reason that the facial shape is potentially diagnostic is this combination of high polygenicity and the ubiquity of pleiotropic effect. There's a high probability that some gene doing that when perturbed produces some metabolic disease. It's also going to have an effect on the face because of how common pleiotropy is in developmental systems. But the third thing that makes facial shape variation useful is a little bit different, and that is the high dimensionality of facial shape variation as a trait. And so this is just an animation here that shows the first 15 principal components of facial shape variation in humans. These components explain a very large proportion of the total variation in facial shape. I can't remember exactly what the number is, but it's the vast majority. And if you think about the number of genes that are potentially contributing to facial shape variation, all of these genes are collapsing onto 15 or so different principal components of variation. So that's an interesting fact in its own right. But the bottom line is facial shape is multivariate, which means that a genetic variant that's associated with disease can not just cause a magnitude of deviation, but also a direction of deviation. It's that direction of deviation that makes facial shape diagnostic. And contrast that, for example, to stature. Stature, equally polygenic as facial shape, but it's a one-dimensional trait. People are either, they vary in how tall or how short they are. We know that stature behaves kind of like facial shape in terms of the first two criteria, and we've tested this out with our database here, because we know that diseases associated with growth and skeletal development have a greater tendency to produce variation in stature than other diseases, and yet not diagnostic. Most of these diseases cause a reduction in stature, so someone is slightly shorter than they might be expected to be. That's a very little diagnostic value to the majority of diagnostic diseases, because it's a unidimensional as opposed to a multivariate trait. They can only be short in one way. Facial shape variation can vary in all kinds of different ways. So we built up this database, and this is work in collaboration with Ophir Klein and Rich Spitz, Francois Bernier, and others over the last, I guess it's 8 years now. We have a database of something like 7,000 individuals More than 700 different syndromes are represented, about 113 of them with sample sizes over 10, which is sort of the threshold that we choose to analyze them. Prefer to work with samples of 20. We have maybe 70 or 75 or so syndromes with samples of 20. And this is the distribution of our subjects in terms of age, and also in terms of self-reported race and self-reported ethnicity. These are the NIH categories for these things. So these syndromic effects, these mutational effects for these mutations associated with genetic disease, are significant in terms of their effect on overall facial shape. And so this is an analysis here, not of individual people, but rather of the mean effects of these different syndromes on the shape of the face. And I've put in here in red, just for, I guess for scale or for comparison, the differences, the mean differences between different superpopulations of humans. And so you can see here, I mean, those differences are actually smaller than you might expect as well. But the syndromic effects are much larger than, say, the differences in mean facial shape between different populations of humans. And it turns out that we can use this information to classify people to syndrome. This is just showing the first pass of this. We published in 2020. We've done subsequent refinements of this, where we get something like 78% balanced accuracy in terms of classifying people to syndromes. And in this particular task, we had 70-some syndromes. I should say, don't take these numbers too seriously, because as you increase the number of syndromes, the classification accuracy goes down, because then you end up confusing syndromes that are more similar to each other. So these numbers of how accurate you are, they're highly dependent upon the exact context of the classification task that you're doing. Right? So there's no absolute number here. Just to brag a little bit, Francis Collins, the former director of the NIH, this was, I believe, his first non-COVID related tweet in 2020 when he saw this paper. For whatever reason, he was interested, and so we were really happy with that. And of course, he had to point out that this was NIH funded, which it was, but we were happy to see him take an interest in this work. So it also turns out that the relatives of people with genetic disease differ from the background population. And this is kind of interesting. The relatives have increased phenotypic variance, and they have a slight but non-random tendency to differ in the same direction as their syndromic relative. So they don't classify to the syndrome of the syndromic relative with a particularly high probability, but they have a higher than random chance of getting classified to the same syndrome as their affected relative. So that's kind of interesting, because these are people who don't have the mutation. Right? So just shelve away that fact, because I'll come back to it a little bit later. Let's just skip over this. Determinants of classification accuracy, if you're interested, the main determinant, it turns out, is actually not the phenotypic effect, how large of an effect on the face the syndrome has. It's rather the distinctiveness of the syndrome. How unusual is the syndromic effect compared to others? So for an example would be Treacher-Collins and Nager syndrome have very large effects on the face, and yet they're often confused with each other because they're similar. And this is also a fact to shelve away, because it's unusual that syndromes in this high dimensional potential space of facial shape variation would tend to have similar effects, right? Okay. So I told you that there's an elevated variance in individuals with syndromic syndromic effects on the face. So what is the nature of this increased facial shape variation? So here again is that animation showing the different components of facial shape variation. And the question that we asked with this is, do syndromic effects follow these same axes of covariation, or do they go in different directions? Are syndromic effects unique in terms of their morphospace directions, or do they just follow the background variance covariance structure? And you understand the difference between these two alternatives? Why might they follow the background covariation pattern? Well, there's a concept I need to introduce here if you're not familiar with. It's a concept of integration, or morphological integration specifically. Morphological integration is the tendency for traits to covary because of some underlying developmental determinant. Great example of this would be the constellation of features that you see in a particular genetic disease or a mutant of major effects, the pattern of pleiotropic effects that you see. For example, in achondroplasia, you have reduced stature, but you also have this interesting and correlated constellation of features, a relatively bulbous forehead, a reduced midface, and increased cranial base flexion. This results in these deep-set eyes that Peter Dinklage uses to great expressive advantage as an actor. He has in many ways some of the typical features of achondroplasia, and these features co-vary with each other. So the degree of penetrance of achondroplasia in an individual is correlated with the extent to which they have these deep-set eyes because of the hypoplasia of the midface or the bulbous forehead because of the increased flexion of the cranial base. The thing is that there are multiple processes in development that do this, that produce these sets of correlated effects. And these processes they act at different times and scales in development. Each of them tends to produce some pattern of correlated effects. So I talked here about achondroplasia. These are effects on cartilage growth. But you can also think about different processes. This is not a complete list, obviously, for craniofacial variation, but you can imagine the processes like this are each driving patterns of covariation in the overall shape of the face or the skull. So there are multiple processes. They act at different times and scales in development, and so they have this tendency of overriding each other because they'll interact in complex ways. So this is what I call the palimpsest model of covariation. So we've tested this model for how covariation comes about in development in a variety of ways, and I'm not going to take you through all of these stories. I have many of them. We could talk about it for quite some time, but I'll talk about the first one because I think in many ways this was at least the one that I was most proud of. This is work that we did in collaboration with Ralph Marcuccio and Nathan Young. It was the first work that started that collaboration, and it was done in chicks. When I say we, I mean Diane Hu was a technician in Ralph's lab, did this work. She took chicks and she treated them with sonic hedgehog and with 5E1 hybridoma cells to depress sonic hedgehog signaling. And we ended up being able to relate together variation in sonic hedgehog here on the x-axis to craniofacial variation here on the y-axis, showing that there was this nice nonlinear curve here relating sonic hedgehog expression to craniofacial shape. And the variation that we got looked like this. We do better animations these days than we did in 2010, but this is now work that's 14 years old. The thing I want you to notice here is that we modulated sonic hedgehog, and the sonic hedgehog is expressed here in the frontal nasal ectodermal zone. But the effects of the change in sonic hedgehog were not localized. They were not localized to exactly where the sonic hedgehog was expressed or even to the mesenchyme underlying it. Rather, there was a global transformation of the overall shape of the face. In other words, there was an integrated set, a correlated set of features that altered when we changed sonic hedgehog concentration in these embryos. So this was a way of getting at the mechanisms underlying continuous variation in morphology. Okay. So do these syndromic effects follow the background covariance structure, or do they go off in different directions? And it turns out, and I'll show you this couple of slides to hopefully convince you of this, they mostly tend to follow the background variance covariance structure. Here's just an analysis of the vectors associated with the syndromic effects. And here are the first few principal components of shape variation, and you can see that the syndromic shape effects tend to align with the first few. They tend to vary most along the first few principal components of variation. Then as you get to the lower principal components, they sort of converge, where it's basically noise. So in this analysis, it shows nicely that syndromic effects tend to follow these first few principal components. Here's another way of looking at this. So I've taken here the syndromic means and projected them onto the principal components for the background variance covariance structure. And you see here that these syndromic means plot along these principal components with these extensions, these elevations at extremes. These are particular syndromes that vary to a particularly large degree along these principal components. And I should say we tested also the relationship between the variance covariance matrix of these syndromic means in the background populations with Mantel tests and random skewers and some other methods, and they're very, very similar. So that actually is an observation that I think is remarkable, because think about the genetic complexity of the face. You have thousands of different genes that are affecting the shape of the face. When I say that the matrix correlation between the syndromic means and the variance covariance matrix of the face is 0.88, I'm also saying you can capture the variance covariance matrix of facial shape variation using, in this case, about 76 different mutations. Right? You only need 76 out of the thousands of different things that could affect the face to capture the overall variance covariance structure of the face. So mutations of major effect in humans recapitulate the background variance covariance structure. It turns out this is also the case in mice. So we have a large collection of mouse mutants, and we've studied this over the years. So this is just an example here showing rhizopathy mutants. And the reason I'm showing you this slide is not that rhizopathy mutants are special in this regard, but they follow the first, and it turns out the fifth, but let's ignore the fifth, but the first principal component of overall craniofacial shape variation in mice. These rhizopathy mutants all form a gradation along the first principal component. And this is true for many different series of mouse mutants. So first principal component of overall shape variation, you take a bunch of random mutants and they tend to align, separate along that first principal component. Sometimes the second, sometimes the third. But again, it's the same thing we're seeing in the syndromic effects on humans. These first few principal components of variation in facial shape are the same directions of variation that these mutational effects are aligning with. So to study this at a genetic level, my student, David Aponte, spent some years modifying a method that Philip Mitteroker, who's here in Vienna, developed called the multivariate genotype-phenotype mapping method. He developed it for a two-way cross of mice. We adapted it for the eight-way diversity outbred mice. And basically what this method does is that it treats genetic variation as a multivariate trait, and phenotypic variation also as multivariate, and it uses a partial least squares ordination to find latent variables that link together the matrix of genetic variation to the matrix of phenotypic variation. Okay, so it's a way of identifying regularities in this underlying mapping of genetic variation to phenotypic variation. This is not a method for finding individual genotype-phenotype associations. There's no method in here that would give you, say, a way of testing the statistical significance of an individual SNP association with phenotype. It's a way of getting at regularities in the overall relationship between a set of genes and a set of phenotypic traits. Okay. And we find here that sets of genes that are associated with different processes. For example, this is genes associated with chondrocyte differentiation. Here are genes associated with the FGF pathway. They correlate significantly with phenotypic variation. There's a sensible latent variable that emerges. When we compare these to individual mouse mutants that have mutations in those in the same pathways, they have effects that often align remarkably. For example, this is FGF pathway, and here's an FGF10 homozygote mutant. The correlation in vector directions between the effects of this mutation of major effect and the FGF pathway overall is nearly 0.7. So, and something about matrix correlations, it's hard to get high matrix correlations, so that's a good result. Sorry, about vector correlations rather. When you look at the relationship between randomly chosen processes associated with different GO terms, we run through sets of genes associated with different genontology terms, and we associate them with the principal components of variation in mouse craniofacial shape, we find that there are many processes that have high correlations with the first principal component, a somewhat lower set of correlations with the second, slightly lower with the third, and then we have fading into noise after that. So multiple processes, at least identified as identified by genontology terms, coalesce on the first few principal components of craniofacial variation in mice. Okay. And these patterns are correlated with each other. I won't talk about that. And when you look at the overall mutational effects for the different mouse mutants in our database, we find exactly the same pattern that we saw in humans, that these mutations of major effect basically recapitulate the background variance covariance structure. So that's interesting. What I think this means, and this is maybe where maybe somewhat overreaching of the original title of the talk, is the issue here. What I think it means is that there is an effective dimensionality reduction as genetic variation gets translated to phenotypic variation by development. So genetic influences funnel through developmental effects. That space is really complicated. At one level, of course, there's a dimensionality increase in development because development is composed of many more things than a DNA sequence, for example. But as that process happens, as genetic effects converge through developmental processes, the effective dimensionality or phenotypic variation is reduced. Many genes converge on few processes, and they produce correlated sets of effects on phenotype. Is at least a feature that we see in craniofacial variation. I think we see similar effects in many other organismal contexts. For example, the fly wing. I don't know much more about Drosophila, but you guys all studied Drosophila, so I won't wade too far into that territory. So if this is the case, then it led us to re-examine these syndromic effects. So if many different genes or many different mutations can push variation in the same direction, what does it mean to have a large-scale mutation having a big effect on, say, the shape of the face? We asked the question of if we take these syndromic effects and treat them as complex traits and map them, find the genes associated with them, what do we get? So this is work done in collaboration with Hilda Peters and Peter Claes at Leuven, and our student, my postdoc, Hannah Hoskins, who did a PhD with Peter, is leading this work along with Michiel, who's working with Hilda. And we've done this now for multiple syndromes, but I'll tell you about the achondroplasia story because I think it's probably the most interesting one. So here we've treated achondroplasia as a trait. And by that I mean we take the difference between people with no achondroplasia and people with achondroplasia. That defines a multivariate vector. That vector becomes a trait that we can project onto the background population, and that trait we can map for genes. Here's just the distribution of individuals with achondroplasia, obviously normalized to the number of individuals. And here's the distribution of people who don't have achondroplasia. And this is the variation, in this case, the cosine distance. You could also use the canonical variant axis, is the trait in question. And so we map this trait and we find all kinds of genes. Sorry about the mess up here. So we get a list of genes associated with this axis of facial shape variation that really represents the achondroplasia phenotype. We do a gene enrichment analysis of this. We find that there's lots of things that look like they're associated with skeletal development, with chondrocyte differentiation, cartilage development, which is kind of encouraging. That's what we expected to see. And you just did and Michiel did this randomization to see whether the genes that we were seeing were more enriched for these terms than other genes that are also associated with facial shape. And it turns out, yes, they are, particularly when it comes to skeletal system morphogenesis. There's a tendency for greater enrichment in this area than for other genes associated with facial shape variation. In other words, treating achondroplasia as a trait, we're recovering all kinds of genes that are plausibly mechanistically related to achondroplasia as a disease. But this is in individuals who don't have achondroplasia. Okay, I know it's a bit of a head scratcher, but you can follow me, I think. So here comes the kicker, though. So we then took these genes that we associated with the achondroplasia axis in humans, and we ran them through David Aponte's multivariate genotype-phenotype model in mice. And this is the loadings of the top loadings for genes that we in this model. And here is the phenotype that we recovered from these genes. And I should note, we did not include fibroblast growth factor 3 receptor, which is the causative gene in achondroplasia. We did not include that in the model. So running these achondroplasia GWAS genes in mice, we get an achondroplasia-like craniofacial phenotype without FGFR3 receptor, which is the causative mutation for achondroplasia. In other words, achondroplasia as a phenotype is something that development is capable of producing in the absence of that mutation. There is a polygenic basis for that potential to produce that phenotype in mice. Okay. The GWAS was on non-disease individuals. That's right. Yeah. Okay. So anyway, this shows you. This also compares this to FGFR3 null mice, which exist. Okay. Similar kinds of things can be said about other diseases. So this is sexual dimorphism. This is a sexual dimorphism axis in humans, and here are genetic diseases that involve possibly a perturbation of the sexual dimorphism axis, and they also tend to non-randomly align to the sexual dimorphism axis. When you take these syndromic effects and you project them into the background population, and you ask the question, well, how much variation do they explain in the background population? You see some interesting patterns. So the genes that explain the most variation, so Fragile X, achondroplasia, hypochondroplasia, growth hormone deficiency, I'm not so sure about the others, but at least these, they're genes, they're mutations that are affecting processes that we think have a good chance of driving a lot of integrated variation. So Fragile X, affecting sexual dimorphism. Sexual dimorphism explains 7 to 9% of total facial shape variation in humans. So big axis of variation, you perturb that axis, and you would explain a lot of variation. Chondroplasia affects cartilage growth in the skull. And so you perturb that axis again, and you can explain a lot of variation. Hypochondroplasia, growth hormone deficiency, pseudoachondroplasia are mainly disorders of growth. So growth, if you know anything about allometry tends to be the first principal component in any morphological trait. So you mess with allometry, you can explain a lot of variation. So these genetic disease effects are channeling onto processes that have a plausible basis for explaining a lot of variation, and they do explain a lot of variation when you project them onto the background population. When you look at this across all these different diseases, you see this pattern here, which is that there is a positive relationship between the severity of the syndromic effect and the proportion of variation that it explains. So syndromes that have large effects, like achondroplasia, tend to explain more facial shape variation. But you also see this covariance distance relationship here. So the larger dots are syndromes that have a bigger effect on covariation, so that covariance structure. So what this tells me is that syndromes that have a large effect, but they have a large effect that follows the background variance covariance structure, they also tend to have axes that are explaining a lot of background variation. Whereas a syndrome that may have a large effect, but it's doing something completely different, something that doesn't actually normally happen, doesn't normally vary in the background population, explains less variation. Okay. All of these findings are consistent with this idea of genetic variance is funneling to a limited set of processes to produce correlations among a constellation of traits. Okay, I don't know how many for time. So as we're doing this, I was actually was a conversation with Charles Roseman that reminded me of this. We had done previous years before, a few years before that, this analysis of genetic and environmental effects on growth in Tanzanian children, but also on craniofacial shape in Tanzanian children. We were interested in this case in growth retardation due to malnutrition. So this was actually work we were asked to do by the schools who participated in our study. They wanted to know whether the kids in their school were malnourished or not, and so we did this study and another one as a consequence of that. And we found that the genetic and environmental variance covariance matrices are highly correlated. And that makes sense in light of this, because again, if developmental processes, in this case growth, are funneling genetic variation, they would also funnel environmental variation. So you have these environmental genetic perturbations acting on the developmental system and then producing a correlated set of features in response. So in Tanzanian children, the G matrix and the E matrix are incredibly highly correlated, like 0.98 something. Okay. Oops, I did something wrong here. There we go. Okay. So this is I don't know if you've heard of Chevreul's conjecture. This is Jim Chevreul, a friend and collaborator who came up with this idea in 1982 when he was working on rhesus macaques, where he found that the G and the E matrices were highly correlated, and he said, well, obviously this is because of developmental structuring of variation. And I think Jim is right. It probably doesn't apply in all situations, may not apply to traits like fecundity traits, but certainly seems to apply pretty broadly to morphological traits. It's also a bit reminiscent of work by this guy here, Per Albrecht, who in many ways was a madman of evolutionary developmental biology in the 1980s, early 1990s. He wrote this really provocative paper called The Logic of Monsters, Evidence for Internal Constraint. But basically what he was arguing is that if you look at the morphology of embryonic lethal mutants, they're not random. They're not just weird things. There's a logic to them, and that logic is underlain by how development is capable of producing variation. And Albrecht's model was something like this. This is also from this paper. So he thought of development as a system, and you could have perturbations on that system. Those perturbations could be genetic, they could be environmental. But the system would respond in certain ways. So it's less important whether the perturbations are genetic or they're environmental. What's important in terms of what the response is the nature of the developmental system and how it produces those phenotypic changes. So this is one way of thinking about how developmental systems respond to perturbation or how genes might influence phenotypic variation. If we think a little bit about the question of what does it mean to say that achondroplasia as a phenotype occurs in the absence of FGFR3, you could even take this a step further and say, well, in what sense is FGFR3 a cause of achondroplasia? Obviously, it's a cause of achondroplasia. Everyone who has achondroplasia has a mutation in FGFR3. I'm not disputing that. What I am saying, though, is that the developmental system has the potential to generate a phenotype like achondroplasia in the absence of that mutation. So that does change a little bit our thinking about FGFR3 receptor gain of function as a cause of that phenotype. Because as a cause, it's an underdetermined cause. You need to have the developmental system potential to generate that phenotype for it to have that genotype-phenotype association. A good analogy for this, I was reading a book actually on Michaela Pavlichev's recommendation by Donella Meadows, who's a pioneer in systems biology. And she used this example of the slinky toy when trying to explain systems to her students. And so she would take a slinky toy. This is a great, I should have brought a slinky toy because there's stairs here. We could put the slinky toy down. And she asked the students, why does the slinky toy do that? And someone would say, well, because it's a spring and it just does that. And someone else would say, well, it's gravity, you're going down the steps. And someone else would say, well, it's because you pushed it. Obviously, it's all of those things. So the slinky toy is a system. It does what it does because it's a system. So is a mutation the thing that makes all of this happen, or is it the push that makes it start down the stairs? And development is the topography of the system that makes the behavior play out. I'm asking that question not knowing the answer to it, but I think it's another way of thinking about mutational effects in contexts where there is a developmental propensity to produce those effects in the absence of a mutation.
You can produce other mutations of large effects that give the same phenotype but are associated with another gene.
Yes, there are many examples of that.
Yeah, so big
yeah, yeah.
So basically, you can have target many genes.
Yeah.
You get the same phenotype, but with a different.
Or a similar phenotype. Yeah, it's hard to say it was exactly the same phenotype, but yes, you can produce. So you can produce a developmental perturbation via different mutations that would produce roughly the same phenotype. So again, it's another way of looking at the funneling of effects.
It's not a single gene that has a major effect in perturbing the system. So you use many genes with similar.
Yeah, that's true. But if you think about the question I was asking about FGFR3, so the standard question is, how does an FGFR3 gain of mutation cause achondroplasia? So you follow the mechanistic chain of events that goes from the gain of function mutation to the disruption of growth of cartilage. The question that you might ask if you think of it this way is, what is it about development, particularly about cartilage development, that makes it so sensitive to FGFR3 gain-of-function mutations? That's a different question, right? It's a slightly different question, but it leads to different experiments. Okay, I want to skip over my rant about the fallacy of gene as a regulator, because I want to talk a little bit about this last part before we're done, and that's maybe an appeal to start getting at these complexities of genotype-phenotype maps by trying to delve into the black box that's in the middle, trying to understand what these processes are that are producing these correlated effects. And I wish I could tell you that we've solved that problem, but it's a really complicated thing to do. What I will do is give you an example of work that we're doing in this regard. So we've been working on ways to quantify cell proliferation or cellular dynamics in three-dimensional embryos and relate that to morphology. We've developed methods for quantifying gene expression in 3 dimensions and relating that to morphology, using imaging approaches that allow us to apply our morphometric methods. And I'll show you some examples of recent work here that we're actually just about to submit for publication, where we used light sheet microscopy to create an imaging registration and analysis pipeline for analysis of cell proliferation in 3-dimensional context and relate that to morphology in individual embryos. And it's the individual embryos thing that's important, because there are many other developmental genetic studies, of course, that quantify cell proliferation in a group of embryos, and they take another group and they quantify gene expression, and a third group and they quantify morphology, but you can't relate any of those things together in that way. So you can't investigate the mechanistic basis for variation among individuals if you don't measure the same thing in individuals. So this is really important, being able to quantify variation in 3 dimensions in individual embryos for multiple things: gene expression, multiple genes, transcriptomics, cellular dynamics, morphology in the same individual embryos. That's what we're pushing towards. And I'll show you some results of the cell proliferation analysis. So we were trying to relate spatial variation in cell proliferation inside the mesenchyme of the forming face to the 3-dimensional morphology of the face. At one level, the work was disappointing because we found out that cell proliferation is surprisingly homogeneously distributed throughout the face. We were expecting to see more stronger patterns. On another level, it's quite informative because it tells us 2 things. First of all, there is a correlation between the 3-dimensional variations in cell proliferation and facial morphology. So there must be some effect, but it suggests that there are other things going on. So probably cell polarity in interactions with the well, actually, I won't go into the range of alternative hypotheses because there's too many of them. I'm starting to get myself confused. But there are other things going on other than just cell proliferation. But the other thing is these embryos are growing really rapidly, and so maybe you don't need a lot of difference in the rate of cell proliferation in different locations to produce a change in shape. And maybe a 1 or 5% difference in the rate of proliferation in one part of the embryo compared to another is sufficient to change the shape that you're going to get if the cells overall are dividing really rapidly, if growth is happening very rapidly. So I think actually that's probably more of what's going on, because when we do relate, again, using partial least squares here, morphology to proliferation, we see that there is, in the early stages of face formation, as well as in the later stages of face formation, there is a clear pattern where spatial variation and proliferation is related to morphogenesis. So it's doing something. This is early work. We don't know exactly what it is, but it is an example of how we're trying to get at these developmental process variables that are driving integrated variation in the shape of the face. So with that, I'll make my conclusions. I hope I've convinced you that mutations of major effect can recapitulate the background structure variation, and that this represents directions of variation that may have a polygenic basis, and so they can be replicated without these mutations. And this is maybe a bit of an ask, maybe particularly in this room, but I think we need to start thinking about the role of genes in development in a different way, rather than thinking of them as I guess, privileged causal drivers of outcomes, thinking of them as contextualized within the broader system in which they produce their effects. This is not to say that genes aren't important. Genes are incredibly important. But maybe we need a different and more nuanced way of thinking about what it is that they actually do in development to produce phenotypic variation. So with that, I want to thank you for your attention and invite any questions.